please join me in welcoming to the Distinctive Voices podium Dr. Matthew Becker.
Thanks. And I know many of you are probably wondering where Akron is. It wouldn't be the first time. Akron's actually about 25 miles south of Cleveland, so I'm suffering a little bit today. Many of my friends are suffering more, but I can tell you that this morning on the flight from Cleveland to Denver, I sat next to Pete Rose, and he looked a hell of a lot worse than I felt. So, so tonight, this is a real treat because this is a really historic place, and my PhD advisor gave this same lecture here many years ago, and she said it was a real treat. And so tonight I really have two aims. One, in the first part, I want you to open your mind maybe to a little bit about more what materials can do. I work in plastics, okay, which is automatically a negative connotation, okay, but I really want you to think about what plastics can do, because probably this decade and most likely next decade are the age of materials and soft materials. The second part of my talk is really going to focus on what additive manufacturing and 3D printing can do with materials. Because again, these are going to be very synergistic relationships between materials and the ability to tune materials and make materials into things that have never been made before. Now, I really tried hard on the flight out here to cut out many of the nasty photos I have. What I do is in medicine. Much of it is military medicine. It's messy. It's dirty. People throw up in my graduate class every year. Okay, I got rid of all of those photos. Okay, I know it's a little after dinner, but that the intent for showing those is not to say that wow, this is neat. It's that wow, these are real problems. Okay, and so and I'll warn everybody that if you've got a soft stomach, when I tell you to look down, you should look down. Okay, because we're not gonna lose anybody tonight. And so that's really the goal. Now, I've been at Akron now 7 years. Prior to Akron, I worked at the National Institute of Standards and Technology in molecular imaging, protein preservation. We worked a lot with stabilizing anthrax vaccine after 9/11. In 2009, I moved to Akron really because I wanted to get back in making materials. I'm an organic chemist. It's a lot more fun to make things and measure things. If you look at my group broadly, we're really focused on how do you develop chemical methods, so I'm a chemist by training, chemical methods where you can place peptides, proteins, bioactive species on materials in such a way that that activity is preserved. Okay, it sounds easy. It's very difficult. We work in a lot of different areas. I've got a group now of about 30. I think I have 22 PhD students, a pile of postdocs and undergraduates for entertainment. Mainly because when I wanted to get in a graduate lab or a research lab when I was an undergraduate, nobody would take me, and so I take special exemption for that. So we work a lot in degradable materials. We'll talk a lot about this tonight. I won't talk about our work in hydrogels. Again, most of our hydrogels, we make materials and send them to biologists that do biological measurements because they're not very good chemists. We make a lot of functional thermoplastic polyurethanes for medical devices. That's work I never get to talk about because the companies that pay for that are there. But know that this is a big problem, okay? About $4 billion in cost to Medicaid and Medicare is due to catheter infections every year. And many, many people die. And so we spend a lot of time and are really making some pretty significant process and really innovation in this area. We also work in nanofibers, mainly for neural regeneration. You'll see the holistic picture of what I'm talking about in the next slide, which you should look down if you have a bad stomach. And really then, how do additive manufacturing take advantage of these unique materials? Okay, in a way that people haven't done previously. So if you have a bad stomach, look down now because I'm going to tell you about the problem we're trying to address. So take about 45 seconds. Okay. This is what an improvised explosive device will do to an arm hanging out of a Jeep. Okay. When I saw this in 2008, it was stunning. Okay. And it's still stunning. These are gunshot wounds, and you might imagine that the prognosis for these isn't very good. Okay, this young man lost his arm. These limbs were saved. This is a small caliber bullet. This is a large caliber bullet. There's all sorts of medieval devices that have been around since the 1960s that they're working to try to address these injuries, but they're not very good. Okay, they work great for civilian populations. If you're in a car accident, you crush a bone, they fix it. It takes a long time, 6 or 7 surgeries. When your arm's hanging out of a Jeep and you pull it back in and you're missing half of it, the outcome's very different. Okay, you can obviously see the bone, but there's a lot of vascular damage. The membrane that covers the bone is gone. A lot of soft tissue, muscle, nerve, blood vessel, and schmutz is what the military surgeons call it. And they cut this off. And since 2001, while there hasn't been many deaths comparable to past conflicts, the number of wounded in action is really stunning, and that's not even thinking about what they're talking about with traumatic brain injuries that we're going to deal about for the next half century. Okay, so, okay, look up. You're okay. It's a big problem. And I show that not, again, to not gross people out, but to really think about it's a very different problem than what most clinicians deal with in the hospitals, emergency rooms, clinics that you might visit on a daily basis, okay? And so when they say, we've got this problem solved, I show them that picture. Well, maybe not. It's very different. Now, this problem does show up in the civilian market, and the reason that there hasn't been a lot of progress on that side, probably in the last 30 years relative to other things that have happened, is that the economic drivers haven't been there, okay? So there's about 2 million bone graft procedures in a year in the United States. Only 200,000 are what's called critical size. A critical size defect is a defect of size of about 3 centimeters that won't heal on its own. You have to graft it or do something or amputate. The clinical indications really aren't very good. This is a lower human limb, a lower human leg. Here's the fibia, here's the tibia. You can see this is a non-malignant, non-ossifying carcinoma. Cancer, it's benign, it's not malignant, but you can see that it eats away at the bone. This is an amputation. If you catch it early, actually this happens a lot in teenage girls. You see that here you might actually do a clean cut, you put the 2 bones in very close proximity to each other, and a couple of things, you can have a couple options. And one, you've got a leg shorter than the other. Okay, they can plate and screw it and you're out of the hospital in 3 days. You can put on an elicit apparatus, and this again has been around since the 1960s, and the resolution's a little poor here, but you can see they've got a closed 2 millimeter gap here. They have concentric rings that go around the limb. They have rods that connect those, and then through the concentric rings they have titanium pins that screw through the skin and into the bone, and every couple of weeks they go in and they stretch this. Okay, and you might imagine that doesn't tickle. I've seen them do it, and I can assure you that it doesn't tickle. It works. Okay, they can grow back inches of bone if the soft tissue there, and more importantly, if the membrane, the periosteal membrane that covers the bone is intact. Okay, all of the blood supply, all of the growth factors that make bone healthy come from the membrane that covers the bone. If you ever split your chin or cut yourself very deep, or you're at the yard with my kids, like we seem to be more than average, you've seen that membrane. Okay, as long as that's intact, you're very likely to have healthy bone. The problem is that humans in general are dirty. Okay, so you could go 2 years, you grow 2 inches back, and all of a sudden you could create a skin infection of any kind. It travels right down the titanium, into the pin, into the bone, and you have a life-threatening infection, and they'll cut it off immediately without your consent. And for all the things that people talk about, autograft, allograft, harvesting bone from your pelvis, femur, there's not enough competent bone to fill gaps like this. Okay, and it's a big problem. Now, the orthopedic surgeons, they solve this all the time. These guys are woodshop butchers. They make it work. But in the polymer space, which is the really opportunity for regenerative medicine, and what I'll talk about in the second half of the talk, is it's been waved off. It was waved off in the 70s. So my father-in-law, who recently retired, he just turned 70, practiced 37 years. He couldn't tell you what a polymer structure was, but he could tell the difference between nylon and lactic acid sutures just by tugging on them. Okay, one will strain, harden, and degrade. The other one's there forever. Same thing your seatbelts are made out of. And the problem is that these were developed in the late 50s, early 60s as degradable sutures, okay, and they became grandfathered in prior to the FDA's regulation on how things were governed for patient safety and they became the go-to materials by which it drove innovation cycles in the industry. Okay, very limiting. These are multi-billion-dollar polymers each, so I don't want to make them think like they're not success. But when you're developing a construct that you want to use in a patient, in a car, in a television, you have to understand what the properties at the end game are and back into a material that works for you. When you start with a limited material subset and try to go in the other direction, rarely does it work. Okay? And yet, time after time after time, big pharma, medical device companies, drug delivery systems, they all start with the same subset, and they end up at the same place, and then it fails. Okay? And so, by and large, these have been waved off. Now, the reason they don't work is that they are semi-crystalline. So this is caprolactone, and I won't test anybody. This is lactic acid. So lactic acid is a natural body metabolite that builds up in your body when you exercise, and that's what makes you sore. They have well-defined tensile strengths, mechanical properties, degradation time. But for what we're trying to do here, the degradation time is too long. And because they strain, harden, and crystallize, you get parts that are amorphous, which degrade faster than the parts that are crystalline. And therefore, you have a real challenge in trying to balance those mechanical and degradation properties during the healing process. Okay? Caprolactone screws, if you've seen a caprolactone screw, they still do this in kids because they don't like to dig them out. If you put a caprolactone screw in a kid in his bone, it'll still be there in 5 years, even though it's a degradable material. Okay? If you put it in somebody who's 50, it'll be there when they die, even if they live to 150. Okay? Lactic acid, same thing. You get some inflammation. And while they were putting in plastic plates and plastic surgery in famous people a long time ago, there's a lot of inflammation, and so for the risk analysis here, which in the medical device community is very low, they just waved it off. Okay. Now, if you go back and talk to even what they're teaching orthopedic surgery residents, which most of the biomaterial community continues to ignore, you really only need two things to heal a fracture. You need absolute fixation. It needs to be rigid. Okay, that's why they use metal plates and screws. And you need blood supply. Okay, and the blood supply doesn't come from the marrow on the inside of the bone. It comes from the periosteal membrane on the outside, which is why it's such a big problem in the military with a blast injury, because all that membrane disintegrates. Okay, and again, this is my PG-rated picture of a bone fracture, but after you fracture, you form what's called a hard callus or a soft callus here, in an anabolic phase. So immediately after the fracture, if it's not displaced, you can see that the membrane here in white is intact. A lot of blood comes in here. You form a clot. Over 10 to 15 days, you form a soft, unmineralized cartilage. And if you've ever sprained or broken a knuckle and you have a fat knuckle, that's what that is. Okay? Over time, if you are biomechanically correct, it will shrink and shrink and shrink. Okay? Unless you have arthritis or rheumatoid, which is a persistent inflammatory state, which basically traps it at this point so that it never shrinks. Okay, so you get a hard callus, and then at about 21 to 35 to 45 days, you recover about half the strength, and then over time, then that's remineralized. Okay, but really, only two things are needed: fixation and blood supply. And so, right after I moved from the federal government, after I saw those injuries at Walter Reed, we really started working hard on developing new polymers that were strong and degradable. We started, and one of my main philosophies in my group is rather than make really exotic things that then you have to stick them in an animal and try and figure out what they degrade into, which is really hard and expensive and painful for them and for us. We really look for, can we find metabolites in normal biological processes, re-engineer those into monomers that you can make polymers out of, so that when they do degrade, we don't worry about so much what happens to it, because we know what those metabolic pathways are. These are an interesting class of amino acid-based polyester ureas. Again, I won't expect anybody to remember this other than these are, in red, alpha amino acids, the building blocks of your proteins that are already in your body. The blue here are simple diols and trials. Again, the terminal oxidation product is a diacid. And urea, again, which is, most of us pee out every day. These materials are strong because they have a very strong hydrogen bonding network that's enabled by the urea. And the thermal and solution processing can be controlled by what type of amino acid is, what the molecular weight is, and what type of diol you might have here. So the library space is enormous. Okay, and we've literally made hundreds of these materials. When they degrade, you get amino acids, CO2, carbon dioxide, readily metabolized diols. Okay, all of which are excreted quite readily, and it's easy to prove. After the fact, after many years, we found that there's a self-neutralization effect. So like polyesters, which I showed you in the last slide, this is the most labile bond. This one breaks first. You form an acid, which leads to some of the inflammatory properties caused by the other polymers. However, you have an acid here and you have a urea-type base, and so it's a self-neutralization, what we're calling a self-neutralization phenomena. Because we've been in 6 different animal models, vascular, bone, neural, never seen inflammation, and that's where we're left at, and we're trying very hard to prove that. And again, very synthetically flexible. The nice thing is the chemistry is not really exotic. So I like to call myself a chemist, but I never call myself a chemist when we show this because this is kind of embarrassing. We take amino acids you can buy, diols you can buy, and under acidic conditions, you can do this in a 5-gallon bucket. And in fact, when I got tired of buying 5-liter reactors, which are about $900 a piece, I sent my students to the Bed Bath & Beyond, and they bought induction cookers and 25-liter pots, and it might look like a meth lab. But you can actually make this in multi-kilogram scale on the benchtop, sub-reflux temperatures with a Dean Stark trap. Okay, this is the easiest organic chemistry experiment you ever do. You can recrystallize it in a bucket. You come back and you can use phosgene, which is a little more concerning, or triphosgene in this case, which is a solid under basic conditions, again in a large pot, and you can make this polyester urea, amino acid based. In this case, it's phenylalanine, which is a benzyl aromatic amino acid here, in multi-kilogram quantities. And we've made as large as 10-kilogram batches in my lab. And now this is the same way that they make polycarbonate, lots of polyurethanes. This is called a step growth polymerization. Very common, amenable to multi-ton manufacturing in single batches. The point I want to make here, and I won't make it too long, is that the C6, C8, C10, and C12 just is the length of this diol. And so you can control the mechanical properties, the hydration, and the degradation of this by changing the length of this diol so that you have something that starts to get flexible at 50 degrees C, and this is the material we'll focus on today, and softens to sub-physiological temperature and then sub-room temperature. So something very rigid and hard and tough to something very soft. Now the interesting thing, and this won't be obvious to this crowd, but I'll point this out because this is a lot of things that the materials companies miss, is that TD is called the degradation temperature. When the degradation temperature is 200 degrees above the glass and 150 degrees above the melt, you don't need any solvent to process it. So it can be injection molded and processed in traditional ways. Okay? And so when you're taking a material through the regulatory process, the 2 most expensive things to prove and where most things die is the first thing the FDA is going to ask you is how much residual metal, how many parts per million metal. The second question is how many parts per million solvent. Okay, and those are really, really difficult when you're making large batches. Now, these are actually quite strong. So this is the strongest biodegradable polymer that's ever been reported in a non-filled composite. It's about 3 times stronger than lactic acid. I won't belabor the point here other than I've got a piece and I'll pass it around if anyone cares to see it in a minute. This is the G version of this picture, but this is really our unmet medical need. And so when the military approached my group about 5 or 6 years ago, we were actually group number 6 to be brought onto this project. So the other 5 have been kicked off. They bring us on, and it actually worked. This is a really productive collaboration with Brad Weiner and Mauro Ferrari at the Methodist Hospital Research in Houston. It's one of the finest medical institutions in the world, especially in the areas of regenerative medicine. Now, this is a sheep model, and I'll forgo the sheep jokes, although there's really good sheep. I have a brother-in-law from New Zealand. There's amazing sheep jokes. Okay. And the military uses a sheep model because sheep are bad patients. Okay? They're kind of ornery. They stand off axis. And the reason they use this is that if you look this is cortical bone and cancellous bone, so hard and soft bone. They have the same ratio of hard to soft bone as humans. And the vascular flux in the limb of a sheep is roughly what you get in a human. This is about a 3-centimeter gap. This is about how much bone you lose if you're shot in the leg with a NATO caliber bullet. And for as long as there's been NATO caliber bullets, if you didn't die due to blood loss, it was almost certainly an amputation. Okay? The military's solution for this is they would like to come up with a single surgery tube construct that would allow them to do what they call limb salvage, save those limbs. And when they brought me on, the surgeons actually took me for a drink, and it was kind of sad, kind of laugh, because these were the 5 things they told me in graduate school. One of the leading surgeons in the world at Washington University, Richard Gelberman, were the 5 things that you could not do for a fracture. Okay, so they wanted to do a single surgery. They want to take somebody off the battlefield in the Middle East, fly them to Germany, which is where everybody who's hurt bad goes, make sure they don't die, do a CT scan, put them back on a plane, 8 hours to Walter Reed or Naval Bethesda, 10 hours to San Antonio, which is where they take most of the people that are burned now. And when they land, have a custom fit piece for that person that they could do one surgery and walk away. Okay? No fixation hardware other than glue. No pins, no screws, no external big rods, fancy things that you see of anybody that's ever been in a car accident, which was, for me, was strike 2. They wanted it to be tailorable to heights and bone structure. So in Cleveland, other than the losers of the World Series, okay, they have one of the finest anatomical museums of natural human history, where they've got a couple of thousand skeletons. So if you go back in most cultures, it turns out that about 3 different diameter tubes would fit 90% of the U.S. military population, all but the largest male, all but the smallest female, for this particular injury. So that was, okay, that we might be able to make that work. Totally resorbable. So someday it had to be gone. The interesting piece here is they didn't really care how long. And that led to a lot of questions, because the paradigm in regenerative medicine and bone tissue engineering, since I was taught in the late 90s, was that you need fixation, absolute fixation. It needs to be rigid. You need vasculature supply. But if you're going to put what they call a scaffold in between where the defect is, where the bone should go around, it should degrade at the same rate the bone forms in. And for polymers, that's impossible. Absolutely impossible, because polymers hydrate, they swell, they mechanically weaken, and then they degrade. And it's at a very different, uncontrollable time scale than you would find in any human. Okay, and the problem is that my son would degrade faster than myself, which would degrade faster than my father. And so predicting that de novo, in addition to the other challenges, is not possible. Okay? And then it's got to be simple. Okay, because I had a really exotic, fancy scaffold the first time we did this that my student, Faye, he worked almost a year on. We took it down to do our first sheep surgery, and the surgeon didn't like how it fit, so he hit it with a hammer, and it cracked, and Faye cried. Okay? And there was not enough beer at Budweiser to get him to continue to work on this project, so I had to get a new student. And so we asked ourselves, okay, if it's got to fit within the confines of modern surgery, okay, which is to even be in an operating room in Cleveland, it's cheap compared to probably LA. It's $1,500 every 15 minutes to occupy the space. That's not the surgeon, that's not the anesthesiologist, that's not the device, that's to breathe in the operating room, okay? So it can't be really exotic. Here was our solution. This is a tube, we call it a shell, because it sounds better than a tube. It's 4 millimeters thick. It's about 6 inches long in this case. It may be a little shorter here. 1 inch inner diameter, which is about what a male of my size, which is too big for health standards, should be. And the reason for this was that we really looked for some inspiration biologically. So this is a cross-section of a human femur. This is a cortical bone. And this is with the marrow, the cancellous bone pulled out. And you can't see the vasculature here because it's small, but basically it's a tube, okay, that provides fixation and blood supply. Now, if you want to actually take our polymer and make this, you can see that we made these are turkey tubs full of polymer that are dried and melt extruded. And I was really fortunate that I was actually at the University of Akron, because this is it looks like plumbing pipe you might find under your sink. That's because the dye from it was we harvested from somebody who was making plumbing pipe from under a sink. But we were really fortunate in that University of Akron is one of 3 institutions in the United States that have a melt extruder this large. Okay, so my colleague tolerated me while we were using it. Otherwise, this project would have never gone anywhere. And that Avram Isayev, I credit, he recently retired. But he was really integral to making this work. Because as I feel like I'm a pretty good chemist, I try not to get too excited. I'm a really bad engineer, okay? So here's my student, Faye, standing on it. You can see it's pretty strong. I've got the piece here, the second generation piece, so I'll pass it around for tours. But in a 4-point bend test, it's about 2,500 pounds per square inch. So I could, this is the piece here, it's degradable. This is the second generation. It's strong. I could stand on it. Try not to break it and embarrass myself. But probably an order of magnitude stronger than it would need to be under normal conditions. And that's where it's exciting. So pass this around if anyone cares to see it. It won't break, don't worry. I would like it back though. So this is the preoperative x-ray. This is the shell insertion here. You see that the periosteal membrane, the white membrane here is intact. We got a 3 centimeter gap. It goes together like Legos. Unlike many other people who are doing bone tissue here, it doesn't go between. It goes around. Okay, and I think there was the clever part of this. It's glued with dental glue. You take an x-ray, you stretch it to the right length, you glue the other side, that's it. You can actually see it here. The key is that it has to be thin enough that you can actually wrap the periosteal membrane around it. And so one of the big challenges working on now with Methodist is where can you get periosteal membrane from? Can you get it from a cadaver? Do we have synthetics? Can you strip it from your shin? How much can you actually take without causing damage to your other bone? But you can see that this entire surgical procedure, while it looks complicated, from the start of the opening incision to when they closed it up, took less than 20 minutes. That's the kind of thing that actually has translational relevance. Now, most exciting was that the last piece that the military wanted, okay, which I didn't tell you in the 5 criteria, which caused the most consumption on the night that they were tasking us with this, was they wanted this soldiers out of bed in 3 days or less, able to put half their weight on that limb. Because they, in their eyes, 3/4 of their deaths were due to depression. Where everything was fine, you've got a young kid, badly injured, we hacked off his limb, everything else is fine, if you can call that fine, and they still died. Okay, so big, big problem. And they thought if they could get them up in 3 days or less, that they'd save a bigger fraction, whatever that might be. And so we were excited that we put this crude-looking splint on here, which is a cast with a piece so that he couldn't sublux. 8 hours after surgery, the sheep could stand up. Okay, 4 weeks later, we cut it off, and it's strong enough that he can run around the field. And if you didn't have his leg marked, okay, or tattooed, what they did, you'd never know you couldn't tell which leg it was. Now, when we did 1-month x-rays, and this was just done last spring, the bone didn't go through the tube, so we didn't look very smart. When you take the x-ray, and then you take it again, and you take it again, and you take it again, why isn't there bone in the middle? There's bone here. You can see that the scaffold's invisible, but there's bone growing around the entire outside, 360 degrees around, because that's the fat knuckle hypothesis I talked about before, and that the blood supply doesn't come from the middle, it comes from the membrane. And so the shell rubbing on the membrane produced enough signal, osteogenic signal, to get bone to grow actually around the tube rather than through the tube. Okay? It took a while to figure that out. But you can see in all the other sheep we did here, same thing. So while there's some mineralization going on, and I shouldn't say the tube's not totally hollow, so it's stuffed with decellularized horse tendon that's on the market for use in humans in Europe, but not yet in the United States. It could be any collagen. All you want to do there is prevent a blood clot, because a blood clot will actually stop the healing process. But you can see in all cases, there's enough bone growing around to support the weight. And in the control here, you don't see that, okay, because there's nothing to hold everything together. Okay, and so at 14 days, this bends, they put the animal down. This was interesting in that on this particular side, which is where they did the incision, the sutures didn't hold, the membrane split. And that the body's normal healing mechanism was able to compensate for that by growing significantly more bone around 180 degrees on that side relative to this side, so that sheep was still able to ambulate normally. Okay. Now this went 4 months. Actually, this was 4 weeks. At 4 months, it's a total structural heal. So now these are the original cut marks, and you can see that this is all new bone. It did grow on the inside. This is new bone. About 40% of the original scaffold volume is gone. There's about 60% is there. At this point, the experiment stopped because we ran out of money. But now I can tell you that with the second generation tube, we just started 100 sheep over the course of the next 2 years to try and see how long it actually goes away, how long it takes for a scaffold of that size to go away. And we have a special military exemption for 12 soldiers if the right patient would present themselves that was injury. And the interesting part is here, and this is the last slide for the first part of my talk, and I'm even about right on time, which is good, is that this is a cross-sectional histology picture, and I'll try to go through this slow. Here's the original glue mark. You can see we fractured it here. This is new bone. This is new bone. This is the hard bone in blue, which is staining the collagen. This is called a callus. You can see the membrane, if you look closely here, this dark red, highly vascularized piece here. If you broke your knuckle, you cut it off and you cut it in half, it'd look like this. Okay, and so the moral of the story here is that there's nothing special about this material. There's no stem cells, there's no drugs, there's no growth factor. This is plastic. Okay? The cleverness here, it's not my cleverness, it's my surgical colleagues, is that rather than try to fight biology's normal healing mechanism, if you can provide a solution where you have fixation and vasculature, so blood supply, and stay out of the way, okay, the body will heal itself. Okay, and that's I think is what's really exciting. Now, we have lots of other crazy things going on with these materials. We're making vascular grafts for a human clinical trial in children with Nationwide Children's. We're doing 3D printing using fused deposition modeling, which I talk about, and wound healing. But I won't talk about today. So that's my materials piece. So I want you to think about materials differently than you might have in the past. The next piece is now, how do you take what you learn in materials and overlap that with what's now becoming available in additive manufacturing? Okay, because that, for me, that's the really exciting piece. Now, I'm giving a lecture on Saturday that's a lot more technical, but this has been way overhyped. Okay, there's lots of challenges with 3D printing in the medical space, especially for patient-specific stuff. So, for anybody who's got an elderly parent or had a CT image or had a CAT scan, wildly accurate medical imaging technology has evolved in the last 20 years. And now it's at a price point where you can actually, they do it too often, right? They give you too many x-rays compared to what you should probably have. The problem for 3D printing, and I'm talking about patient-specific 3D printing, is that it's easy to do the image. And then you get to this, what's called a design phase. So you have to design a part for you. Now that's, again, contrary to how medical devices have evolved in the last 30 years. Okay? Because at the FDA, they look at what's called, and all companies have it for every device they make, what's called a design history file. So as they make iterations or improvements, even if they're very small over time, they've got benchmarks, data, mechanical engineering, imaging, manufacturing protocols that go all along with that in a design history file, okay, which includes the material. What they're proposing to do with additive manufacturing for medicine is to take medical imaging, throw out the design history file, and print something for you, okay? And the piece that is going to, and the FDA is working really hard on that with really smart people, okay, is how do you convert this digital design to something that's printable in a way that would uphold to scrutiny if something goes wrong, right? Because it's all risk management. What's the right jaw structure for you? What's the right leg structure? If your head ends up flat and you're not happy when it should have been round, you know, those are the kind of things, and people are chuckling, but I mean, those are the kind of things that are really, how do you ensure that your design ends up into a print code that's actually what it should be? Chemically, structurally, biomechanically. Because once you have that print code, there's any number of things that can print it, okay? Then it becomes, what's the right material for the person, for the application? And so you really, this whole process is create a part using engineering drawing software, and this is where a lot of the IP and innovation access is really ongoing right now. Trying to correlate and leap from medical imaging to high fidelity printing that's biomechanically sound, okay? And then patient anatomy can be accounted for via the imaging, and then the porosity and internal reinforcements can be added if they're needed. There's lots of exoskeleton stuff out there, spinal cages, easy things. So this is not really new, okay? So right now in the United States, there's 85 different things that are 3D printed that are used in humans today. And they're adding about 3 a month, okay? So it's not new. My father-in-law is an oral craniofacial surgeon, and his 3D printing, it's a little cruder, right? But 15 years ago, 20 years ago, you'd come in, you'd get a dental mold, okay? They'd send it out. Somebody else would print it off-site or mold it. Come back, does it fit? No, it goes back out. Now they're doing this in their offices, okay? Why? Because they're temporary and they're support materials, so they're never going to stay there. And so the long-term risk is low, and in oral craniofacial surgery, it's a payer's market, right? So there's not a lot of medical reimbursement, either from Medicare, which controls most of medical innovation, or insurance companies. Now, the biggest market's probably patient-matched surgical guides, again, in craniofacial knee and ankle, where because of training rules and how the 80-hour work week for surgical residents, okay, a lot of this patient-specific guide, they take a medical image, they print a part, and it teaches them where to drill the hole when they don't have line of sight. Okay, so you should always ask who's doing your surgery when you go in. And it's nice to get the attending and not the resident because they know where to drill the hole. Okay, lots of other things, cranial implants, pseudo-custom knees, spinal cages, things where there aren't really good solutions. So these are going on. Now, this is not what I'm going to talk about today. It's much more attuned to the military piece, and I got rid of all the bad pictures for this because they're even worse than what I showed you before. So this gentleman was in a motorcycle accident, okay? Skid marks, cracked skull, orbital bone, nose, mandible, jaw. Not a very good prognosis for this person. They don't take bone from your hip or your leg to fill this. What they're doing now is actually they cut out the small bone in your leg. And so again, this has been now the standard of care probably for about the 15 years, and I'll show you why in a minute. But they can take this, and the reason they use the small bone in your leg, again, is it's nice and straight. Okay, it's about the right size for your face. If you look like Heidi Klum before, you're not going to look like Heidi Klum after anyway, so that's less of a concern. But it has the periosteal membrane, and the blood supply is intact. Okay, and you don't need that small bone to get on with your life. If you're a marathon runner, you have bigger problems. And what they do is they can actually design in silico how they might chop this up so that they can fill huge gaps in your face, either in the lower jaw or in the upper mandible. Okay. Now, the next one's a little not quite as squirrely looking, but I think I'll try to be careful here. And so if you've got a large fracture injury here and they want to model this, they can actually take that bone, which is green, and print a surgical guide here, which is basically a cut guide, where you see the blue corresponds to the blue, the light blue, and the dark, and the yellow is the throwaway piece. But if you cut correctly, okay, it folds very nicely into the right anatomical structure for your jaw. Same thing for the mandible. Here's the printed device here. Again, that's not quite as elegant as this one, but here it's basically router, cut. And you can see it here actually in work. They lay it on the bone, chop. You can see the intact blood supply here. When they cut this out and they stick it in your face, they graft the blood vessels into the existing blood vessels that are left in your face. And in only a couple of weeks, you've got competent bone that heals that you can come back and screw teeth into. Okay, which in a tissue-engineered construct would take months and months and months. And so, again, you might not look very good, but you're eating, breathing, things that are essential to life. I'm missing a picture here because this is an older one. Again, this is a model of Congressman Gifford's skull from Arizona. So she was shot in the back of the head, and the bullet came out just above her left eye. This is the mirror image. This is actually the picture here. I left the other picture out. But NASA, okay, and this had only happened at NASA, took my colleague's technology, David Dean, who was at Case Western at the time, is now at Ohio State in plastic surgery, pirated his technology. NASA 3D printed out of dental resin a skull for her, put it on her back for, I think, 6 weeks, and then came back and actually attached it to her skull. So she's got a non-degradable plate in her skull. And if you've seen a picture of her lately, she looks pretty good, okay? And so this is what 3D printing can do for a patient-specific piece. Now, is this a large market? No. But how this works is actually pretty interesting. And this will maybe demonstrate what some of the challenges are before I get to the chemistry. So when you've got a neurocranial defect, which is basically a hole in the head, the gold standard is an autograft. Can you get enough bone or pelvis if it's small? In a big hole like this, you get back to Cousin Eddie, in Christmas Vacation where he said government plastic. It doesn't look all that great. Now, in doing this, it looks easy, right? The cartoons make it look easy. But in fact, when you're coming back with polymer, even if it's non-degradable polymer, you've got to design either through the mirror image or the anatomical structure of the face how that part might fit. Okay? And the margin for error is small. Because again, if you want the skin to heal, and you can't put pressure on the brain, but you can't put pressure on the skin, or the skin over the top of the scalp will die if the polymer swells. Okay, so you can actually take an anatomical structure and do an averaging, and you can see it's sectioned in layers because that's how it'll actually print. Here's the paper here, and you can see that this is more than 13 years ago they were doing this. And I think David Dean, when he was at Case Western in neurosurgery, actually did 350 patients. And so the technology is very vetted, it's very there. Now, you will have plastic in your head the whole time. This doesn't heal, it's still plastic. But the thought process in this, and how would you actually get it to heal? Or why would this even make sense? Okay, and it'll make sense here in a minute. But you can actually map how you would do this, and if you shave it off, you can look at the overlap regions. And you want to obtain about 50% overlap, again, so you don't put pressure on the bone and kill the existing bone where it contacts, but it would actually heal and the skin would fit over. And why this makes sense and why they actually do this and how it actually came to be is that at Case Western in Cleveland, preoperative fabrication saves OR time, which is, again, $150 to $300 a minute. Okay? Your plastic surgeon doesn't do this because you're paying him. The government's not. Medicare's not. If you want to go in, have your nose done, ears done, breasts done, whatever, you pay for that, and they're drawing on you with a marker and making it work. Here you can see that this is a 3D printed cranial skull out of plastic. The skull model's in beige, the draft implant is in red. This is the part they play with and look at the overlap, and the final implant's actually in white. You can print this, sterilize it, use it, and again, it saves tens of thousands of dollars in OR time. Now, all of that's non-degradable. So this transition here, maybe in the last 15 minutes or so. The only current degradable thing that's out there that the FDA is tolerating is actually a resorbable trachea stent. This is being done by Scott Hollister and Glenn Green at the University of Michigan. And Scott, I think, and Glenn both just moved to Emory in Atlanta. But this is a tracheal splint for babies with a collapsed airway called a tracheobronchomalacia. Okay, 100% fatal for all of human history. Okay, now in utero, with a uteral ultrasound, they can detect that this is happening. And you can see they detect it before the baby's born, again, because the oxygen blood supply is coming from different places. They can actually print a model of the trachea, and you can actually see in red here, it's a little tough to see. They print a polycaprolactone, that material I said wasn't great. It's actually pretty good in this case. Stent. So that as soon as the baby's born, they slap them on the bottom and they stick in the stent, and it opens up the airway. 100% save rate. I think they've done 24 babies. Again, this is a pretty rare condition. 24 babies. If they find it anywhere in the country, they get sent to the University of Michigan, and probably now will get sent to Emory. And it's been 100% safe. Okay, it's made on an EOS Formega P100. This is about a $25,000 printer. Probably next year you can get it for under 10 because they've got a new generation on polycaprolactone beads you can buy. And the solution for this is it gets integrated in the tissue, but when the child is 3, 4, 5 years old, the thought process, then they'll go in and they'll just yank it out. And they should be fine, right? So it looks like it's going to heal. And that's really the power of what resorbable materials could be. Again, 100% fatality rate for all of human history, and now they've solved that. Again, we get back to if you want to do other things where you need a little more control over degradation or properties or drug delivery or protein, whatever, you know, that we're back to this same toolbox. And that, for us, that was really frustrating. So I'll skip my nasty photo here again. Okay. And so the three materials that the companies will sell you when they sell you the printers, they'll sell you, there's, I think, a list of 60. 57 that are non-degradable in these three. You get lactic acid, glycolic acid, caprolactone, and they will take you to the woodshed on these. They cost an order of magnitude or two more than everything else, okay? Because the shelf life's not very good, and the printing processes aren't very good, so you've really got to control that. The glass transition properties, the point at which they soften, is pretty high. The bigger problem here is you get asymmetric degradation due to crystallinity. Now, A candidate material that's really been exciting for us, and one that we looked at a long time ago when I was a student, is called polypropylene fumarate. So they've published 1,400 animal studies on this material, and it's never gone to humans because the way that they're manufacturing it is not economically viable and not amenable to FDA's regulatory process. Now, why do people care about this? One, it's got a reactive bond. It's also a polyester, but it's a room temperature fluid, much like honey. Okay, so if you want to have a flow pattern where they do photochemical printing, it's very amenable to that. The reason it's exciting for us, and the reason we chose to revisit this a couple of years ago, is that it fits in with our normal thought process and how we design materials, in that if you cut this ester bond on both sides, this is fumaric acid, which is found in fumarate in the Krebs cycle, okay, so natural body metabolite. And polypropylene glycol, you'll find it in shampoo, but it's also a diluent material in how they press pharmaceutical pills together. So you eat this every time you take anything, okay? Resorbed, it's fine, it's been around 50 years. Now, how they had been making it was a process called step growth polymerization, which is how polyurethanes are made, foams, okay? It's fantastic chemistry for very high molecular weight material. But beyond 3,000 Daltons, which is about 14 subunits of this material, it transitions from that honey material, which is very useful, to a solid, which is not useful for printing. Now, this is diethyl fumarate, very expensive, okay? Propylene glycol, you can tense this under high energy conditions. You drive off ethanol, it generates a lot of heat, and you get very uncontrolled polymer properties. So small molecular weight chains, intermediate, large, it's a mess at low molecular weight. And that's why, even if this cured cancer, you couldn't manufacture it this way at 3,000 Daltons, because in a consistent manufacturing, good, or what's called GMP processing, it just doesn't work. So we spent a little time on this, mainly due to David Dean's insistence that this was a really important problem, and me not being able to get rid of him quickly. We started with maleic anhydride and polypropylene glycol. So these are the two of the cheapest commodity materials there are. They use this to make tires, and they use this to make shampoo. Okay, we were really inspired by Jeff Coates' work. Jeff Coates is at Cornell University. He's one of the leading experts in sustainable materials. He was looking at feedstocks like this for degradable packaging for food and consumer use. Okay. In a two-step process now, so not just heat, we're using a catalytic process and then an isomerization. I'll show you what that is in a minute. Mild conditions, atom economical. What that means is there's no byproducts. You don't have to drive things off. You don't have to add things in. Okay, so for when you're trying to quantify how much metal, how much solvent, you can sidestep a lot of those problems. Very controlled polymer properties. Okay, so this is molecular mass distribution, which is basically a distribution of chain length. And when you have distribution of very tight control over chain length, you can control degradation, which is what we're really after in this process. And more importantly, it's reproducible. Same conditions work at 5 grams, work at 50, work at 5 kilograms, work at 500 kilograms. I'll skip the chemistry lesson for tonight, but here we use a magnesium-based catalyst, magnesium ethoxide. The terminal oxidation product is magnesium oxide. Again, found commonly in all pharmaceuticals as something you can eat, and they press together to make it a solid. And ethanol, okay, which we know what that does. Subcritical toluene, this is a cis isomer, this is a cis isomer, and using a simple amine, and now we're looking at solid state methods. You go from the cis to the trans, you have a fluid, all the analytics are built in. And the glass transition property, if you look at, skip all this stuff and look at molecular mass, this is 700 Daltons, 3,000 Daltons, above 4,000 it's a solid. But you can see the TG here, so this is sub-room temperature, this is Celsius to 12. More importantly, the viscosity properties. And so the optimal viscosity properties here are about this 0.005. This is deciliters per gram intrinsic viscosity. That means it's about the consistency of honey. And you can drop it. You can go to high molecular weight if you heat it and drop it. Now, how 3D printing works photochemically. And again, this is about as simple as you can make. You have a resin bath here, which is basically a pool of honey. You have a tub. From the bottom, you have a light source. So this is the same light source that you find in your LCD television or LCD projector. Okay, projects through a window, transparent window. You have a motorized access. And then you have basically an oxygen depletion zone. So very near the window, you get a focal plane or an in-focus image that will print turn that honey into a solid. And then the motor sucks it out of the, you can see it here, the motor sucks it out, and so you're growing here from the bottom. So you print here first, and these are printed first up here and less here. Very pattern-wise. And just like you get colors on your LCD television, all sorts of different colors, all sorts of different dyes, wavelengths, lifetimes. We'll skip the chemistry for today, but these are the resin chemistry. You get about half of it is the material, half is the solvent, and then you get some other additives that are initiating substances in developing. So basically dyes that initiate radical that turn the liquid into the solid. So this is a gyroid-type structure, which is the mechanical engineering most robust structure. This is for a rat cranial scaffold prototype that we're doing. It's 12% solid, 8% air. Okay, and so this is what's design elements. Now, one of the other things, and I'll show you this slide because it points out some of the challenges. Because it's a layer-by-layer process, it's inherently weak in one direction. Now, there's some really exotic things going on in South San Francisco with a company called Carbon. You might have seen this, where they're actually solving this problem by doing a continuous liquid interface process. They have some resolution images, but I think that's one of the big challenges that's now been overcome. So if you print at 90 degrees or 45 degrees, either print geometry is fine. You can see that they're about, on average, 25 to 30% weaker than the bulk material. But you can compensate for that if you can control degradation. Okay, and that's the point I want to make. And so I've just got a few slides left. I've got about 10 minutes. And so if we look back now at my sheep example, where I said the paradigm for a long time had been, if you want to do tissue engineering of bone, the material has to degrade at the same rate the bone forms. Okay. In polymers before, they hadn't really thought about how to do that. But now that we have a polymer, it's degradable. Fumaric acid, which is resorbed in the Krebs cycle, polypropylene glycol, very short chains, even though they're chemically cross-linked, we might be able to control this if we can control geometry. Okay? So here's two different test structures. This is what's called a fine structure. This is a coarse structure. 6 millimeters across, 10 millimeters tall in both cases. This one has a 200 micron strut. This one has a 400 micron strut. 700 micron pore, 1400 micron pore. Basically, 88, so 12% solid. This one has half the surface area as this one. And so if this degrades due to water absorption and hydrolysis coupled with enzymatic action, then higher surface area should degrade faster. Okay? In this particular experiment, we've got the same material, so 1800 Dalton material. This is an accelerated test, kind of like cheating. Over 30 days, this is mass fraction of material, and you can see, it's a little small here, these are triangles, these are the circles, that with higher surface area, you degrade much faster. Okay. Here we have two different materials, same structures. This is the fine structure. Here we have 2400 Daltons, so a little bit longer chain, more cross-links per unit chain length. And you can see that the lower molecular weight, again, degrades much faster than the one that's higher cross-linked. Most significantly, okay, we don't lose, this is day 0, day 20, day 30, with, I think this is the 1500 fine, 1500 fine structure, 1500 coarse, and then the higher molecular weight, is that there's no defects in the pattern. And so it truly is a surface eroding mechanism. And so you don't get chunks breaking off, which would be counterproductive to your bone healing process. And some of the chemistry is shown here, and if anyone has questions, I'll ask. But the key to this is that if the chains are short, even though you're creating covalent bonds between the chain, as long as this, what I'll call chunk, is left over smaller than 7 nanometers, which is smaller than your human serum albumin, which your kidneys retain, okay? As long as it's smaller than that, it will come out, okay? And anything that's smaller than 7 nanometers will come out of your body through some active or passive process. Now, if we can do that and really control anisotropic structure and degradation, for the first time, you've really got an opportunity to look at how you might plan a segmental defect repair. So this is a dog model that my colleague David Dean and I are working on at Ohio State, where you can cut out a section of the mandible and actually, with the mirror image, project how you might grow that back. Now this is our early structure, right? So it's basically a homogeneous gyroid, which again is the most mechanically robust. But you can imagine that if you can control anisotropic or complex patterns, you might be able to have areas where the outside of this structure would grow faster than the inside. okay, or based on the stress patterns, you might have struts in here that weren't homogeneous like the gyroid. Okay, so there's the in silico design, and I can tell you that this actually does work. And so here's the in silico design. We've got a plastic part here, but it's well defined enough that you can actually predict where you might want to tap the screws to use metal plates on a first pass, but then actually use plastic or fully degradable sections on the end. And I can tell you that in the dog models and in the, I guess, the artificial bite things that we're using, even though this is only 12% solid, it's actually strong enough to create enough force that this could potentially chew. Okay, and so with that, I think the rest is just more complicated. So I'll stop there. We're doing a lot of experiments. It's something that we're pretty excited about. I think, and again, the goal tonight was not overwhelming with chemistry or gross fixtures, but really, if you're thinking about materials, there's a lot of room to innovate in materials. There's a lot of companies now starting to develop pathways where they might bring new materials to market, and I think ours is a good one. It's probably not the best one, but I think looking at polyester ureas and especially taking it from a degradation standpoint and that we know what those degradation byproducts are going to be is interesting. We've got a number of applications. It's highly tunable. The buffer degradation, I think, is really an emerging success hypothesis. We're desperately trying to prove it because a lot of people have been challenging that. But again, we've been in 6 different animal models in 6 different species and not seen inflammation. And so this has to really be it. But still, we have to prove it. What we're really excited about is this functional healing of an unsupported defect is unprecedented. And we've got a clinical trial teed up hopefully next year. I hope nobody's hurt, but in the event somebody is, my feeling is a year from now that we'll have done first in man with the military. I think the polypropylene fumarate may finally reach the clinic. There's been 1,400 animal studies. In the past 25 years, they've never gone to the clinic, and now we've got clinical trials that we've teed up at Mayo Clinic in Rochester, Minnesota, Ohio State, and Langone Medical Center, New York City. One is an orbital fracture for elderly patients where they don't want to do a hip bone graft. Another one's actually trying to grow back the lower bone in the leg that you take out. And another one is for a cancer model. Very inexpensive precursors, not that it'll be cheap to buy it, but I think the fact that the precursors are cheap actually suggests that there's a translational path to actually get this to market in some reasonable time. But I think really if you're looking at materials and how you might combine that with additive manufacturing to really change medicine, the ability to design asymmetric degradation is what I was taught even not that long ago would never be possible. And I think now, coupled with what instrument manufacturers and innovators are doing in the additive manufacturing 3D printing space, what many groups like myself are trying to do with bringing new materials to the interface of that technology, I think now you're going to be able to see where if you can design asymmetric degradation, they're going to be able to do incredible things for trauma surgery, for reconstruction, and also things that haven't even thought about doing. The patients are waiting. Okay, so the numbers are. I care a lot about military populations. The numbers are small, but we ask a lot of them, and I think they're waiting patiently with some pretty disastrous outcomes. And really, I hit this a lot because when I give talks similar to this at device companies, at pharmaceutical companies, when I get this, that'll never happen because that's not a material that exists on our list. I sell their stock if I have any. And I tell them that, look, tomorrow's problems will not be solved by yesterday's materials. And I really believe that. The people that are willing to put in the time, put in the risk, there's going to be a lot of things out there. It's not going to happen with big companies. It's going to be small companies that get it into the clinic, it actually works, and then it'll be bought by some of the bigger boys. So this is my group. Like I said, I've been at the University of Akron 7 years. Most of you probably, I could count on one hand how many people actually knew where Akron was. The first polymer course, there's actually 2,400 polymer companies and chemical companies within 100 miles of Akron. It's half of all North American manufacturing in plastics is within 150 miles of Akron. I think we have more jobs in plastics, rubber, coatings, automotive than Michigan times 10, and I think as much as the rest of the United States combined. And so it seems a little off the map. The first course was taught there in the world in polymer chemistry in 1909 by Charles Knight, and the things that happened in synthetic rubber, which I noticed were on the hall driving the Beckman Institute. The things that were done at the University of Akron in World War II in synthetic rubber were second only to what happened in the Manhattan Project. And so it's been exciting for me. I've got a really talented group. I think I've got, it's a little unusual in that it's half women. I try to really promote that because I've got an 8-year-old princess who, as she gets older, my hair goes away more. This work was funded by the military. Okay, when I first submitted this proposal to NIH, they told me I was throwing my career away. Okay, so you got to be a little stubborn. And when I testified to Congress 2 years ago, I told them that nothing was innovative was happening at NIH, that they should give all their money to the military because those are the people really with needs and where things are actually happening. Now that's not true, but really talented collaborators. The things I'm doing can't be done in a vacuum. Okay, and one of the things reason I think we've been successful is that I own a lot of failures and share a lot of credit. Chris Brewer and Tai, Chris is the only person in the United States doing a human clinical trial on tissue-engineered vascular graft at Nationwide Children's, again in a fatal fetal condition. He's been fantastic. We're making tissue-engineered vascular grafts for him. I think that hopefully is going to go well. The 3D printing that I showed you at the end was pioneered by David Dean. David Dean was putting 3D printed parts in human heads when I was still in high school, and is really a really humble guy, but doesn't get enough credit for really driving that market. Mike Miller's the head of plastic surgery at Ohio State. He came from NYU as well. And then much of the early work with DARPA and the Department of Defense was Brad Weiner, who's the head of spinal surgery at Methodist, and Ennio Toscati, who's his lead PI in nanomedicine there. And again, these are two of the best guys in the country, really humble. Turns out Brad Weiner was born and raised in Akron, and that's how we actually got connected. So with that, this has been a really great privilege. I'll answer as many questions as I can. This is work that's ongoing, and so it's certainly not finished, but I hope I leave you with the two things that additive manufacturing is changing medicine, and it's going to be enabled by materials. So thanks.